Greetings, biochemistry students, and welcome to this PowerPoint lecture about the citric acid cycle. This is Dr. Michael Garut. The citric acid cycle oxidizes acetyl coenzyme A, which has 2 carbons, and this is an overview. In each cycle of the citric acid cycle, 2 carbons are oxidized to carbon dioxide, and 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of either GTP or ATP is produced. This diagram of the citric acid cycle shows the enzyme names of each of the 8 steps. It has highlighted in green the 2 carbons from acetyl coenzyme A, showing that those 2 become these bottom 2 carbons in citrate and isocitrate and alpha-ketoglutarate and succinyl coenzyme A, which means that the carbon dioxides that are liberated in steps 3 and 4 are not the same carbons as the ones in the acetyl group. That means that even though the stoichiometry is 2 carbons enter and 2 carbons leave, the 2 carbons that leave must have come from other citric acid cycle intermediates. This diagram shows, or in this diagram, I'm trying to show the radio tracer studies that were done in order to elucidate that information. For example, if one puts a radio label on this carbon, the carbonyl carbon of acetyl coenzyme A, and then follows the molecules where that is found in a cellular extract with all of the enzymes present, they will find that it's here at the bottom carbon of citrate, isocitrate, alpha-ketoglutarate, and succinyl coenzyme A. When you get to succinate, the molecule is now symmetrical, so now you cannot tell whether the carbon is carbon 1 or carbon 4. They're equivalent, so that isotopic label is now diluted 50% in each of those 2 locations. That carries through to fumarate and malate. Now let's look at the second cycle. So as we continue on from malate, if I now label one of those carbons with this orange label, I now have 50% of that isotopic label that came from the first carbon of acetyl coenzyme A. That one now becomes the top carbon of citrate, isocitrate, and alpha-ketoglutarate, and is liberated from alpha-ketoglutarate in step 4. If you followed through the carbon that was next to that carbon, so let's say this carbon, then that one would become this carbon, the second carbon of alpha-ketoglutarate, and so that carbon is not the one that is liberated. So in the second cycle of the citric acid cycle, 50% of the carbon that was in this original acetyl-CoA goes out. None of this one. That's following it through as far as we need to, just to understand how the experiments were used to determine where the carbons were going in the citric acid cycle. This diagram from your textbook describes the chemistry of each of the steps, so that we have a Claisen condensation and elimination and addition, also known as a dehydration and a rehydration. We have oxidative decarboxylations. We have what's called a substrate level phosphorylation, which is when a phosphate is directly transferred from one high energy phosphate to another. We have a dehydrogenation, which is another oxidation reduction reaction, a hydration, which is an addition reaction, and another dehydrogenation, which since produces NADH, you know that's another redox reaction. This is a good time to introduce or remind you of the common names of the dicarboxylic acids. That is because many of the citric acid cycle intermediates are dicarboxylic acids or are based on dicarboxylic acids. Citrate is actually a tricarboxylic acid, but when you get to alpha-ketoglutarate, it has carboxylic acids at both ends. Succinate, carboxylic acids at both ends. Fumarate, malate, oxaloacetate. All of those are dicarboxylic acids. If you are using the IUPAC rules, for, let's say, glutaric acid. It has 5 carbons. So you would call that pentane dioic acid. But the common name for that has existed much longer than pentane dioic acid, is glutaric acid. The conjugate base of that would be glutarate, and that comes from the Latin combining form gluten, which means glue. It's the same word that is gluten as in gluten intolerance in proteins, which is the glue that holds flour together when you are making breads or cakes. Well, alpha-ketoglutarate is, in fact, that 5-carbon carboxylic acid with 1 extra carbonyl group. In the IUPAC, if that carbonyl group were on carbon 2, you would call it 2-oxo-pentane dioic acid. We can also call that alpha, because this is the alpha carbon, alpha-ketoglutarate. So the 2-carbon is oxalic acid or oxalate. The 3-carbon is malonate, which is not the same as malate. Malate is this molecule. Malonate is the 3-carbon dicarboxylic acid. You have succinate, glutarate, adipate, and pimelate, or adipic acid and pimelic acid. And here's a mnemonic device you can use to help remember those first 6 dicarboxylic acids. In these next slides, the specific chemical reactions of each of the steps are shown. I'm going to go through those briefly, very briefly, because you already saw them in the other diagrams. So in the first step, you transfer 2 carbons from acetyl-CoA onto oxaloacetate, taking a 2-carbon and a 4-carbon carbohydrate to make a 6-carbon carbohydrate. This is the organic chemistry mechanism showing that you have an active site aspartate, which is acting as a base, and you have 2 active site histidines, which are acting as acids. The aconitase step does an isomerization, so you're isomerizing the hydroxy group from carbon 3, which is a tertiary alcohol, onto carbon 2, making it a secondary alcohol, and therefore making it able to be oxidized to a carbonyl group. Step 3 does that oxidation, transferring the electrons to NAD to make NADH, and then decarboxylating to give off 1 carbon dioxide. You might remember from organic chemistry that a beta-keto acid is readily decarboxylated, and that's what you have here. Step 4 is the alpha-ketoglutarate dehydrogenase complex, which is evolutionarily related to the pyruvate dehydrogenase complex, uses all the same cofactors as that complex. We saw how that worked in the last video. Step 5 is succinyl-CoA synthetase, which is named after the reverse reaction, as is commonly the case. A synthetase is a reaction that joins 2 molecules, often using a high-energy phosphate for energy. In the reverse reaction, or the reaction in the direction that it actually proceeds, you are attaching a phosphate to GDP. So it's a free inorganic phosphate that's being activated in the active site and then attached to a GDP to make GTP. That's a substrate level phosphorylation reaction. The final 3 steps are an oxidation, a rehydration to make another alcohol, and another oxidation. So the oxidation in the succinate to fumarate step is taking alkane and making an alkene. That does not provide as much energy or is not as energetically favorable as making a carbonyl. And so the cofactor that is made is an FADH2 because that requires less energy to create. In the final step, we're using NAD again because that is an oxidation that produces a carbonyl. And it is important to note that the succinate dehydrogenase complex is also found in electron transport, which we'll see later. Here are the energetics of the 8 steps. The delta G naught primes are the standard free energies of the reactions under biochemical conditions. Notice that steps 1 and 4 are extremely favorable or far from equilibrium. And the other one that is the next most favorable is step 3. So that's the isocitrate dehydrogenase reaction. Those reactions as you might predict, are the key control points for the regulation of the citric acid cycle. One of the things that you want to be able to do when you're studying metabolism is to rationalize why it makes metabolic sense for ATP, for example, to be an inhibitor of the pyruvate dehydrogenase complex, while AMP would be an activator, and all of the other ones except for calcium, which we haven't really discussed yet. The anaplerotic reactions will become important later on in the course. Right now, suffice it to say that you can create citric acid cycle intermediates from proteins, but you cannot create them from fats because the fats come in as acetyl coenzyme A. That means there must be carbohydrates in the form of oxaloacetate for that to react with in order for you to be able to metabolize the acetyl coenzyme A. And in each cycle of the citric acid cycle, those two carbons are used up, so you're not increasing the number of carbohydrates available from fatty acids. Citric acid cycle is also the central hub of metabolism, serving as the starting materials for many other biomolecules, which we'll see later in the course. And a consequence of the fact that acetyl-CoA cannot be used to increase the concentrations of the citric acid cycle intermediates, that means that since oxaloacetate is the starting point for gluconeogenesis, or if you consider pyruvate to be the starting point, it's step 2 of gluconeogenesis. That means that animals which lack these enzymes that we're looking at here do not have the ability to convert fatty acids, which break down into acetyl coenzyme A, into glucose. Plants and some bacteria do have these other two enzymes. What these two enzymes allow plants and other bacteria to do is to basically increase the number of citric acid cycle intermediates by converting 2 intermediates into 3. Let's take a look at how that works. If the acetyl-CoA comes in, when it gets to step 4, where isocitrate makes alpha-ketoglutarate, if instead of doing that, where you start to cleave off the CO2s, you have this enzyme, isocitrate lyase, that produces a 2-carbon carbohydrate called glyoxylate and a molecule of succinate. So you have bypassed the 2 steps of the citric acid cycle that liberate carbon dioxide. If you do that twice, now you have 2 2-carbon molecules, 2 glyoxylates, and you could hook those 2 glyoxylates together to make a malate. Typically, however, malate does not use 2 glyoxylates, but it uses 1 glyoxylate and another acetyl coenzyme A, which is a different 2-carbon source. But you take 2 2-carbon molecules and make this 4-carbon malate out of it. So from 2 molecules of isocitrate, or 1 isocitrate and 1 acetyl-CoA, depending on how you think about it, you get 2 molecules of succinate and 1 molecule of malate. That allows you to increase the concentrations of the intermediates of the citric acid cycle, and since those intermediates are used to make other things, such as glucose or amino acids, that allows plants and some bacteria to be able to make those from fats where animals and humans cannot. That's the end of the lecture about citric acid cycle, and in the next one we'll look at the electron transport pathway. Cheers!